Hello, everyone, and welcome to the 30th day of the 8th International Meeting and Plant Breeding. My name is Igor, and without further ado, it's my great pleasure to introduce our distinguished speaker in the field of agriculture and environment, Professor Rodrigo Mendes. Professor Rodrigo Mendes holds a bachelor degree in agronomy and a PhD in genetics and plant breeding, both from Exalc USP, as well as postdoctor from the Van Hove University and from the Lawrence Berkeley National Laboratory. Throughout his professional career, Professor Rodrigo Mendes has acted as visiting scientist at Canaviales in Monsanto, the University of Lausanne, Rothamsted Research in the United Kingdom, and the University of Cambridge. He served as a head of research and development at Embrapa Environment from 2015 to 2022, and is a member of the Scientist Committee for the Promise Research Program. Currently, Professor Rodrigo is a researcher at Embrapa Environment and a professor in the graduate program at the University of São Paulo. His research focuses on soil and plant microbial communities, aiming to elucidate how the Rhizoesphere microbiome influences plant growth and protection. We are excited to hear his perspectives on this important topic. Please join me in giving a warm welcome to Professor Rodrigo Mendes.
Hey, good morning. Thank you for having me here. I'm happy for this opportunity to share with you the concept of the second plant genome, as if the plant genome itself was not enough to make our lives complex. But when I give this talk to farmers and growers, I start with the following question. So what if you decide for any reason to grow your crop for 10 years in a row without adding any fertilizers? What if you do that for 20 years? So they quickly say, so my crop is gonna collapse, they're not gonna grow. But what if you do that for 50 years without adding any fertilizers in your fields? The good thing is that someone already did that. And I'm gonna share with you this experimental data. With this experiment, it started in 1845. It's from maybe the oldest experimental station in the world at the Rothamsted Research. And for more than 150 years, they have been doing these experiments. This is one of the experiments, the broad bulk experiment. There are two points here that I'd like to highlight. First, the reason why we were able to increase the productivity was the combination of the introduction of new techniques, as following liming, herbicides, fungicides, the use of NPK, rotation, combined with what we have here in the axis, the introduction of superior genotypes. So we need the combination of these two things to increase productivity. And this basically sustained the growth population in the world. Here in the 50s or 60s, we have the Green Revolution, and then we have a bunch of new technologies coming in, and this was able to sustain the increase of productivity. Technologies with genetic material. But there is a hidden layer in this graph, which for me is really intriguing, which is the control treatment. So in this light blue line, there was no fertilizers used. They not even used farmyard manure. That was one of the treatments that they have in these experiments. But even so, the wheat was able to grow in the field for more than 100 years. Of course, the productivity is much lower when we compare with the other treatments, but even so, the wheat could grow. So what is sustaining the growth of the wheat over these 150 years in the field? So what is going on below ground with these biogeological biocycles that are able to give to the plant what the plant needs to survive and to grow. And this is what I would like to discuss with you. And maybe this is the answer for the first question. So even without adding anything, there are something below ground able to help the plant to survive and to develop. To go even in early years, this was the device that somehow opened our eyes to a new universe. So do you have an idea what is this? No, this has about 5 centimeters. It's a small. Yeah, this is one of the first models of a microscope. This was invented in 1685. So there is many researchers working on that, but this model was created by Anthony Leeuwenhoek. And this is where you have the lens. The lens, it's about 1 or 2 millimeters. And this is where they put the sample, and they use a candle on the other side, and they started to see cells, microbes, plant cells. But can you imagine it? So the first blood cells, the muscle cells, they didn't even use this name at that time. But can you imagine when Anthony Leeuwenhoek took a sample from a lake, from a pond, and for the first time observed in this microscope? What did he see? What did he see? Microbes. For the first time. They called them animalcules. They didn't use the name microbes at that time. And then he started to sample environmental samples and describe and draw all these small animals, protozoa, bacteria, structures of fungi for the first time. And they somehow opened a new universe. But then, 350 years later, a new microscope was invented. And do you recognize this one? This is a sequencer, a DNA sequencer. It's interesting to look back the microbiology history, because everything started with environmental microbiology, when you see the microbes in the context of the environment. But then for 150 years, we didn't have any much progress on these studies. But then on the early 90s, we have the gold age of microbiology. But at that time, we used it to study single microbes grow on plates in the lab. So we removed the microbes from the context of the environment and the complexity of interacting with other microbes. But then we have the second microscope, and then we have the chance to, again, to see the complexity of the microbiology in environmental samples. So when this came out, so a key point in history was when we did the human genome sequencing. And this was in the beginning of this century. But just after we finished to sequence the human genome, we realized that to understand the functioning of the human body in a complete way, we need to take into account the human microbiome. And then a new big project started called the Human Microbiome Project. And one of the first papers on Nature that published about the human microbiome, they have a figure showing a draw of a human with more or less 23,000 genes, plus more than 1 million genes that are from the human microbiome. So then you have to take into account all these genes working together. And this is the concept of a superorganism, or a holobiont. So taking the host and the microbes together, we see it as a unity of evolution. So it changed the mindset, the way we look an organism. And of course, we can transfer that to plants. I will get there to the plants. So, but one of the impacts of the use of this technology was that we reorganized the tree of life. So we, in the 90s, using molecular data, we divided the living beings in the world in 3 big domains: bacteria, archaea, and eukaryota. But since then, when we used the second microscope, we have been discovering new microbes that had changed the topology of the tree of life. For example, you see this big branch here. This branch is called candidate phyla radiation. All these microbial lineages lack on representatives. So we don't have them on freezers in the labs. We only have them on computers. So we are not able to grow those microbes. So when we use sequencing to look environmental samples, we discover new microbes. We cannot have access to them in the lab, but we know the DNA sequencing. And as we add those microbes in the tree of life, we are changing the topology. And this keeps changing. This is from 2006. There is, I think, another paper restructuring this part of the tree. But what is the message? So using this new microscope, we are discovering a bigger diversity of microbes. So and we didn't know that they exist. And of course, we didn't know what they are doing when they are associated with plants. But what we did learn over these last 15 years when we started to apply this technology to study microbes. So first, we know better how to count microbes in samples. And this is a parallel with this human microbiome figure that I mentioned to you. So the range of variation is huge in plants, but if we assume a host with 40,000 genes, and then we look at the rhizosphere. So I'm going to define later what rhizosphere is. But if you look only to bacteria, a single gram of soil sample from the rhizosphere harbors maybe up to 1 billion bacterial cells. And each cell has around 6,000 genes. So the number of genes of bacteria living in close contact with the plant is much bigger than the number of genes of the host. And if you take into account the other members of the community of the microbiome associated with plants, then we can easily increase the complexity of the system. So first, we know better how to count the diversity. And we keep increasing because we keep discovering new microbes. We also know better the functions of those microbes. But traditionally, we know the good microbes. So if I ask you here, and you tell me some beneficial microbes associated with the plants, you can easily tell me. Nitrogen fixers, phosphorus solubilizers, and so on. And if I asked you if you can tell me which are the bad guys, bad microbes, the pathogens, we can easily list a long list of pathogens. But these are the extremes in the spectrum of the diversity. In between the bad ones and the good ones, there is a universe of microbes that we are still discovering. And some researchers, they call them biological dark matter, that is still are there to be explored. But even we are not aware of the existence of them, they're playing a specific role in association with the plants. So then the study of microbiology in the last years somehow moved in a direction of understanding the whole of microbes living in complex communities, instead of looking at the single isolates. So let me introduce you the concept of disease-suppressive soils. Now we're gonna dive on a small plant experiment. Disease-suppressive soil, by concept, it's a soil where you have a pathogen in the soil, you have a susceptible host, but you don't have the disease expressed, or you have just a limited amount of disease in the field. The counterpart of a suppressive soil, as we call a conducive soil. So basically, those two soils are from the same farm, they share the same physical and chemical properties, but for some reason, when you grow sugar beet seedlings in both soils and then you introduce the pathogen, in this case it's Rhizoesphere solani, in this case we don't see disease, but in the conducive soil you see the damping of symptom. The fungus is able to grow in the soil and colonize and kill the plant. So this suppressive soil was described, again, more than 100 years ago. The farmers in Australia, in Washington State, United States, looking at the wheat and the pathogen Guaianomyces tetrici, they observed that after a disease outbreak, if they kept cropping the wheat in the same field, after 3 or 4 or 5 years, they observed a decline in disease. The pathogen was still there in the soil, but the disease was gone. What we did was to use the second microscope, sequencing, to revisit this phenomenon. And this is basically the data of this experiment. So, of course, we suspected that the explanation for this natural protection of the plant was due to the microbes, and then we can basically do 2 tests to reveal the nature of this protection. So the first simple test we did was to sample a suppressive soil with low disease incidence, and then we take the soil. So if you take the suppressive soil and then we heat treat the soil for 30 minutes at 80 degrees, we transform a suppressive soil in a conducive soil. And this is what we have here. Suppressive soil, heat treated, and then we repeat the experiment. The experiment is growing sugar beets, and then we introduce the pathogen. So when we heat the soil, when we heat treat the soil, we kill microbes, and then those microbes cannot protect the plant. So this is the concept. Yeah. And the other simple test that we can do with suppressive soil is the soil transferring. So if we take a suppressive soil and then we mix into the conducive soil in a 10% ratio, we're able to decrease the disease incidence. When we do the soil transfer, we are transferring the microbiome. We are transferring microbes and also molecules and compounds and genomes and introducing in the conducive soil. And somehow the plant can communicate with those microbes, and then those microbes can help the plant to fight against the invasion of the fungus. And this has many parallels with the gut microbiome. And I have these, so basically there are some striking similarities on how the microbiome interacts with the host. And the way we see the rhizosphere is that the rhizosphere for the plants is like the gut system inside out. So between the host and the environment, there is a living layer of microbes playing a key role on many important functions, including protection against pathogens, production of enzymes and antibiotics, the modulation of the immune system, and also a list of other functions that the hosts rely on the microbiome to survive. So basically, when you look at the plant, we can have 3 assumptions. So first, we see the plant as a superorganism. So we take the genome plus the genes and genomes of the associated microbes. So second, the host relies on the microbiome for some specific functions. So the host can use outsourced services provided by the microbiome. And in nature, we have many examples on that. For example, ruminants, they rely on genes present on the rumen microbiome to break down the fiber that they use. And the third assumption is that the host is able to some extent, to modulate the assembly of microbes associated with itself. There is some level of genetic control on which microbes can be associated with me and how those microbes work and functions. So if you take this view of these three assumptions, we can get this model. So basically, we used that small experiment, and then we used many different approaches, including amplicon sequencing, metagenomic sequencing, or metatranscriptomic sequencing to see which genes are being expressed during this natural protection. And this is the summary of this story. So basically, when we introduce a seed in the soil, the seed, when it's germinating, it starts releasing some chemical compounds, attracting some specific microbes to be associated with the plant during the development. But this chemical communication is also realized by a pathogen. In this case, Rhizoesphere solani, that it's is leaping around, realizes the presence of these specific compounds and then germinates and then starts growing towards the root systems to infect and to kill the plant. But when the pathogen gets close to the root system, there is a reaction by the plant. There is a modulation in the physiology of the plant, and the plant changes the metabolic exudation profile in the soil and causes two things. First, increases the abundance of some specific members of the microbiome in the rhizosphere, and activates some genes in those specific microbes. In some of those genes, like genes producing antibiotics, they are able to protect the plant against the invasion of the pathogen. So basically, when the plant is under stress, the plant changes his physiology and asks help to the microbes, and those microbes come to help the plant. This is the hypothesis, as we call the cry for help. And this is not only for protection against pathogens, but we can use the same rationale for example, abiotic stress. When the plant is under stress, a lack of water, drought, the plant modulates itself, changes the release of chemical compounds, and attract microbes able to help the plant to tolerate the stress. And this is also true for nutrients. When there is a lack of phosphorus in the soil, the plant communicates with the microbes, and microbes related to, for example, phosphate solubilization helps the plant to get access to the nutrients. So looking at these model, I would like to highlight 4 important strategies to deal with this, or to learn from this model and apply in production systems. So first, microbial diversity is important. When we compare suppressive soils with conducive soil, we see that suppressive soil, they have a higher microbial diversity. And now you can understand why. Because if the plant cries for help, if you don't have those microbes with those functions that are needed by the plant will be attacked or will suffer. So what is the problem with the conducive soil? The plant is trying to call beneficial microbes, but they are not there. So when you look at the production system, it's really important to have a high microbial diversity. So one other strategy that we learned from this model is that when the plant reacts to the invasion, there are some specific beneficial microbes that are enriched in the rhizosphere. So what if we identify those microbes, and then we grow them in the lab, and then we apply in the soil? So this is basically the most common commercial strategy used nowadays, the use of inoculants. For these experiments, we did the same. We isolated hundreds of microbes, and then we identified some key microbes that when we reintroduced those microbes in conducive soil, they were able to protect the plant against the invasion of the pathogen. So a third lesson from this model is that we always think of adding beneficial microbes in the field, but actually the communication between the plant and the microbes, it's chemistry. So maybe we don't need to add new microbes. If we trigger all these reactions with specific signals, this could be a strategy that we could use. So for this specific experiment, we also demonstrated that, because when the rhizoc is getting close to the root system, there is a oxidative stress, and the fungus produces acid oxalic. And then we thought, so what if we use a conducive soil and then we add oxalic acid? Could you have the same, can we trigger the same response from the plants? And then we showed that we are able to do that. So adding oxalic acid in a conducive soil triggers similar processes, and then the plant can be naturally protected by the Rhizoesphere microbiome. And then the fourth one is that, and then this is, now I'm talking more to breeders here. Who is governing all these interactions below ground is the plant, is the host. So maybe we could have plants more efficient in orchestrating all these beneficial interactions below ground. And the question is this true? If you think that the plants that we have today are quite different when we compare with wild plants, when you think of domestication and breeding, we somehow narrowed the genetic diversity of the plants. Maybe we left behind some key genes that could be involved in this type of interactions. Then we tested this hypothesis. So we call this initiative Back to the Roots. So there is a website you can access. Basically, we proposed a framework on how to compare wild plants with modern relatives to test the hypothesis, to understand it. So what was the effect of plant domestication on the interactions in the Rhizoesphere microbiome? So for example, we have an experiment with common bean. The center of origin of the common bean is the Andes in Colombia. So we went to Colombia and we sampled some wild common bean, and then we selected modern cultivars of common bean. We sampled the native soil in the center of origin, and then we grew wild plants and modern cultivars in the same soil under the same conditions to see what is the difference in the recruitment of beneficial microbes in the rhizosphere. So we have, there are groups working on common bean, tomato, potato, millet, and also wheat. And I will finish giving an example on wheat. We use the same approach. We selected some land races coming from Pakistan, Turkey, Iran, and then we select some wheat from old breeding programs in Brazil and some more recent varieties of wheat. And we used that second microscope to understand the pattern and the structure of the microbiome in the rhizosphere of these different materials. So we look at the bacterial community, the fungal community, and also the cercozoa community. So if you will look at for example, this first one here. And then we also use agriculture soil with a history of wheat cultivation. And then we also used soil from a forest in a field on the side of forest located on the side of the wheat field. So if you look at these analyses, the code colors, the blue shades of blue, are all samples coming from modern cultivars of wheat. And the hot colors are all samples coming from land races of wheat. So the first lesson we learned doing this experiment is that when we grow these contrasting genotypes under the same conditions in the same soil, the pattern of recruitment or the assembly of the community, it's different. And it's different not only for the bacterial community, but also for the fungal community. So we can discriminate, looking at the fungus, the fungal community living associated with the plant, we can discriminate, we can tell, so this is a old material, wild material, or a land race, or you can tell, no, this is a modern cultivar. And this is also true for Cercozoa associated with the plant roots. And then we used a co-occurrence analysis or a network analysis to have a photo of the complexity of the interactions below ground in the rhizosphere. So basically, this has many mathematical parameters behind this picture: centrality, modularity, number of edges and connections, positive or negative correlations. But when you look at the picture, we have an idea about the complexity of the possible interactions between all these three groups of microbes living in the rhizosphere: fungi, bacteria, and Cercozoa. So this is the graph for the land races. And when we do the same analysis for the modern cultivars, this is what we get. We missed something here, right? The land race and the cultivars. It's much less complex in this case. So first, in the last years, we know better to what extent the plants rely on the rhizosphere microbiome to survive, when you take into account this cry for help hypothesis. Then we saw that the plants, during the domestication and breeding process, they have somehow the interactions between the plant and the microbes were mined by this process. So we left behind possibly some genes that were important for the plant to get access and to recruit these beneficial microbes in the rhizosphere. And then the piece that it's lacking is that, can we use this approach to go back in wild plants and bring and reintroduce in modern cultivars this ability of the plants to interact with beneficial microbes? And then for some years, some groups have been trying to use the microbiome data as a phenotypic data to link the genetic traits of the host with the recruitment of specific beneficial microbes in the rhizosphere. And then basically the framework for that, and this is a paper from 3 years ago. It's a Dutch group from the Institute of Ecology in the Netherlands. They proposed a model, and it's interesting. So they call extracting the genes. So basically, they are adding the M letter in the model, like this, to treat the microbiome as an independent element in the model, and the G by E model. So if you look at the paper, there is elaboration of this, and it's even more complex. So they justify the importance to bring as an independent element in the model the genes associated in the microbiome and to take into account during the breeding programs. There are other terminologies used by that. So before that, the same group, they used the term MAPS, microbiome associated phenotypes, to deal with the possibility of the microbes to change the phenotype of the plant and how to bring that to breeding programs. And more recently, this year, another group, they use the M gene breeding. M also from microbiome, how to introduce to bring the microbiome to the scene when we do plant breeding. And to finish, I'd like to mention the two key papers from 2 years ago where they used the same approach. It's very similar approach. So basically, those two papers were published back to back in the same issue of Nature Communications. The Dutch group on tomato from Jos Heimarkers, and this key paper is from David Bulgarelli. He's from University of Dundee in Scotland, working on barley. So what they did was to get a wild genotype crossed with a modern cultivar, and then they have some sort of a mapping population, and they were able to link specific traits or KTLs in the genomes of those materials linked with the recruitment of specific beneficial microbes in the rhizosphere. So basically, they paved the way to, as they go, rewild modern cultivars to get the trait of those ancestrals and bring back to modern cultivars, and those cultivars ultimately will be more efficient in get associated with beneficial microbes, and with that they could naturally be more protected against invasion of the pathogens. The plants could better deal with abiotic stress or get access to the nutrients. So to finish, I just listed 1, 2, 3, 4, 5, let's say, a short list of what it's coming in the next 10 years on the plant microbiome studies. So first, exploring the unknown. So we keep discovering new microbes. We keep exploring this biological dark matter. Many microbes that we don't know, we don't have access, but they have some importance in the interaction with the plant. So this will come in the next years. So far, especially in microbiome studies, they are predominantly descriptives. But if you want to manipulate the microbiome to get a better plant performance, we need to understand the mechanisms. And this is also quickly changing to understand so who is there and how, or which are the mechanisms of interactions. So third, plant microbiome breeding. I think there are many groups already trying to incorporating these different approaches to bring back the microbiome interactions during the selection in breeding programs. Trait-based approach, especially microbiologists, we also tend to think about the taxonomy. So the plant needs these specific species of microbes associated with the plant. But the complexity, and especially what as we call functional redundancy in the microbiome community. So we have to skip the name of the microbe and look at the functions, because when the plant is crying for help, the plant doesn't care about the identity of the microorganisms, but the plant goes to the function that the plant needs to be helped. And artificial intelligence. So there are many really nice papers using microbiome data to predict soil health, plant growth, plant performance, and so on. And some tools will also help us to deal with this complex amount of data, and also computational power to deal with microbiome data. That's it. Thank you for your attention, and I'm happy to get some questions.
And now we have time for questions.
Thanks a lot for your presentation, Rodrigo. As you talk directly to the breeders, I felt like, okay, I see that we are maybe missing some things while we are breeding new genotypes, new cultivars, and new hybrids. But how do you see that we can select for more efficient genotypes in a large breeding scale? So talking about thousands of plots. Thank you.
Yeah, this still is a big challenge because getting access to the microbiome data, it's really expensive. Really expensive. Yeah, especially when we scale in a breeding program. So the way we see that is that, so normally we think that when we do domestication and breeding, as we are not aware of the importance of microbiome, we left behind those microbes. But actually, for some specific traits, although we are not aware of the importance of those microbes, we are selecting positively for some specific traits. And one of these examples is the association of common bean with Rhizoesphere. Although we neglected the importance of microbes, we selected materials more that are able to more efficiently to associate it with Rhizoesphere, without no. And then, so not all traits were negatively selected. But then the way to pursue that, I would say, so first we need to think of a characteristic that can be connected with the microbiome. For example, in a breeding program for searching for drought tolerance, that we know the importance of microbes. So in this case, what is the suggestion? Is to get a mapping population with contrasting parents to be able to identify the genetic trait associated with recruitment of microbes that can alleviate the stress. So the idea is to target specific functions, and as we identify those traits, then we use traditional techniques to bring back these traits to modern cultivars. This is so far how we see how to tackle this problem.
Thank you, Rodrigo, for this great presentation. My question is about the microbiome network. You present that it's for the land races, it's more complex than for cultivars. But think about breeding. We want efficiency in process and a lot of metabolic process wastes a lot energy, thinking in yield especially. So we are having less associations between microbiomes and plants, but they are being more efficient, or it's not true?
Yeah, of course, because we can have a trade-off. So when we select it for productivity, and then we can leave other things back. We don't know yet in detail. Our point of view is that the assembly of this community in the rhizosphere is to some extent driven by the genetic of the host. And as the selection pressure we used in breeding selection, we somehow alleviate the need of the plant to get access to nutrients, because we just give nutrients that the plant needs. To deal with drought, to deal with pathogens, because we take care of the plants. And then those genes were out of the genome of modern cultivars. So then the microbes that are associated with those modern cultivars, they are there by, as we call, stochastic processes. So they are there by chance. While when we look at the wild material, there is a deterministic process in the recruitment of beneficial ones. But today it's very difficult to say what is the balance. So should we go back, but then we're gonna get a reduction in productivity. So what is the link between productivity and those QTLs that can be associated with recruitment of beneficiaries? We don't know yet. But this is something that will, I think for sure many groups will shed light on this in the coming years. But it's something to keep in mind. Yeah.
So in the slide with the questions, which kind of traits could you model? What can you put in this left-hand side in the Y term?
So there is a list of traits that are linked with recruitment of beneficial microbes. So I don't know by heart these traits, but they are all traits linked to the physiology of the plant. So how the plant deals with, for example, drought stress. And there are some key compounds that the plant releases when it's under stress, and those traits are linked genetically with those groups.
Rodrigo, currently we see each day more inoculants that they rely on 2-3 species of microorganisms, and we don't focus on the genetic part of the plants. But according to your presentation, we should focus on the genetic parts of recruiting instead of focus on the microorganisms. What are your thoughts about that?
When I look to some data of specific inoculants, there is an interaction between inoculants and the genetic background of the plant. So some researchers, they said that we have to get specific inoculants for specific cultivars. And some people, they say, no, we need an inoculant that it's able to, so because this is the dream of every breeder, yeah? So to not have these interactions between. But we see that in the field. And this is one of the reasons why the use of inoculants sometimes is not so consistent, because we change the genotype and then we change the interactions. Not only that, but we somehow neglect the complex of the microbiome in the soil. Because so for example, when you look at the network analysis, and then we see that complex interaction between microbes, for an inoculant or for a pathogen to invade and to establish in those conditions, it's really difficult, because the inoculant has to compete with other microbes. But if you have a more losing interaction in the rhizosphere, then it's much easier for the inoculant, but also for a pathogen to establish in association with the plants. But this is something that we're still learning. It's important. I think from now on, I think we're gonna be more aware of the importance of the genotype and how to combine that with when we select specific inoculants in agriculture.
First of all, thank you for your presentation. It was amazing. And my question is about the gas emissions. Like, do you think that we can use the data, like with the gas emissions of the soil, to compare with the soil health and use this data in breeding programs?
Yeah. There are some groupings looking at that, because of course the microbes, they are associated with all these cycles in the soil. The way we see is that, so can we have soil microbiomes that helps the carbon sequestration in the soil? And this is also something relatively new, because traditionally people in the models for carbon sequestrations, they are more concerned about the plants above ground. So when you harvest and then the plant goes to the soil and it's degraded and the carbon goes back to the system. But it's also intriguing that plants, this is not really clear, they release in the soil depending on the species or depending on the stage of development, up to 40% of the carbon assimilated in the photosynthesis is released in the soil. So why the plants spends a lot of energy fixing carbon and then releases in the soil? So of course, we suspect that the plant uses this energy to communicate with the microbiome in the soil. But the point is that the carbon that is processed through the rhizosphere microbiome, it's much more stable than the carbon that comes from the above part of the plants. So the microbiome plays a key role in sequestering the carbon and keeping in stable forms in the soil. So then there are two things: understanding the mechanism and the metrics of this. Nobody knows yet. Which are the traits of the microbiome related to the processing of the carbon, and all the traits related to the cycle of the nitrogen. So this we know the basics of that. But I think in the future, we can somehow use efficient plants to manipulate the microbiome of the soil, and the outcome will be less emissions and a better carbon sequestration. I think the microbiome has a key role on all this process. Yeah, this is open to be studied.
On behalf of the organizing committee, we would like to express our sincere gratitude for your presentation and for sharing your expertise with us today. And we would like to offer you a gift.
Thank you. Thank you. Thank you very much.